Okay, so we feed our bees all sorts of types of things, and I guess the question is really, do we understand what we're feeding the bees is doing to the bee health? For as long as I can remember anyways, and I've been doing bees now for half of my life, it sounds a bit scary, doesn't it? There's been the debate as to whether you feed honey, and you only feed your beehives honey, or whether you feed your bees sucrose. So who feeds just honey? Is there anybody in here that just feeds honey? That's the only thing that they are. So there's a few of you in here. Now we also have this thing where invert sugar is coming into play. So maybe we should only be or not using invert sugar. We don't really know. And I guess that's the question we should be asking ourselves. Do we understand what we're feeding our bees and how is that impacting on our bee health? So I've got a small little study that I'm just going to present to you today. It doesn't tell you anything about bee health, unfortunately. But what it will do is give us a bit of an indication as what's happening within the beehive and within the bee gut and what impact that's having on the bacteria that's in there. And it's a good place for us to start, a good place for us to draw a baseline and get a good understanding of what's going on. So just to give you a little bit of a heads up, there is 1 billion bacteria in your gut of your honeybee. So really, you're not actually keeping bees, you're keeping bacteria, really. It's all about the bacteria that's going on in the gut. So what is happening in there? Is that being impacted by what we feed it, or is it not? So we have 1 billion bacteria in there. And we know, what we don't know is that what impacts on that bacteria and what that impact has on bee health. And what we do know is that as the bees are foraging on food and water and propolis, they're collecting a whole bunch of different types of sugars, sucrose and fructose. These things have impacts on bacteria in lab studies. They're also collecting bacteria and the different types of nectars that they're picking up, or antibiotics or pesticides. So all of these things are going into the gut, they're ingesting them, and then potentially having an impact on the bacteria. So what we did is we looked to see what happens when we feed in the lab these invert sugar, sucrose, and some honey on the bacteria that's in the gut. And what we looked at first, we took Manuka honey as the sample honey because we know that it has antibacterial properties outside or just on topical applications. So we thought, we'll give it a go in the honeybee and see what happens. So I grabbed 2 Manuka samples. One's a 2015 Manuka sample, and the second one is a 2017 Manuka sample. They're both from the same apiary. The 2015 Manuka has a higher MGO, so theoretically more impact or more bacteria, more antibiotics. The 2001 had a medium level of MGO in there. I then also used a synthesized MGO and a synthesized DHA, both of which the MGO was at a high level, same as the Manuka level. I made it the same because I had the Manuka tested. And then the DHA I had at about 1,600 milligrams per kilogram. I then also compared a invert sugar straight from a bulk source, so 67 bricks, and then I compared that with a 50% sucrose. Now the MGO and the DHA I had all within a 50% sucrose solution, so they're still getting some sucrose in there as they're feeding. And then I had my controls, which were basically the bees caged and put back into a hive for the same length of time. I used 10-day-old bees. And so these 10-day-old bees I had already marked when they came out of them, they emerged. I marked them and put them back into the colony for 10 days and let them free fly. And then I collected them back up again. So I had to paint quite a few bees with nail polish, so they look really pretty. And then I had to collect them after 10 days and then pop them into these little cages. And so that was great. And then after 10 days, I looked to see what was impacting on the bacteria. So this is just a little graph, and I'll just explain it to you. My treatments are along the x-axis. So I've got my hive, sucrose, invert sugar, the 2 Manuka honeys, the DHA and the MGO. And I've stacked those up, and so each of those colours is a type of bacteria. And the one thing the most important thing from this graph that you can notice is that every single bacteria that is important in the hive sample is also present in the treatment samples. So what this tells you is that you're not going to kill an entire bacterial family by feeding your hives sucrose. So that's good to know. It tells you that the bacteria that's in the gut of the honeybee is fairly stable. So that's another good thing for us to understand. Super stable, seems to be super stable. But we look at this graph and we see it's a stacked graph. So we lose a bunch of information with stacked graphs. So I also looked at my bacteria on a plot like this. And so the idea with these plots is that you look at the colours there, and if there's a clump of blue together, and that's different to say from the clump of red, then you'll know that there's something going on between the different treatments. And you can see here that all the blues are spread out, all the yellows, all the greens, all the oranges, they're all spread. There is no significant difference here going on at all. So this type of plot is a Jacquard plot, and this plot, what it does is it looks at the differences between the treatments. So there's nothing in there. But in my stacked graph, it looked like there was a little bit of something going on with the Manuka honey treatment. So then what I did is I looked to see whether I could, I plotted it again using another different matrices. And this one here gave me some clumping. Didn't clump with the treatments at all, but there's some sort of clumping in there. And this is using unweighted data, but what it also does is it pulls in the taxonomy from the different bacteria. So it pulls in the different types of species of bacteria and it compares them. So I can see that there's something going on, but it's nothing really to do much with the treatments there exactly. So what I have to do is do a little bit more digging to identify what's really going on there. So we just have to wait a little bit longer so I can do my analysis on those, but that'll be coming pretty soon. So just to summarise, gut bacteria is not going to be wiped out by a single carbohydrate source, so you're okay there. It doesn't seem to be any difference between honey, sucrose, and invert sugar. But what that doesn't mean is just because there's no effect there, it doesn't mean that you should feed it to your bees. So really the take-home message is look to see what you're feeding your bees. Do you know what it's doing to your bees? Do you know what it's doing to its bee health? And then make the decision about what you're feeding your bees based on what you actually know, rather than just what you think. So, any questions? My time is up.